Hello, I'm Ed Chapman, and this video cast is going to give you a very quick overview of the animal kingdom and focus on a few of the aspects of this big part of biology that might be important in figuring out some AP biology questions on the AP exam coming up in May. So let's go through this quickly, and hopefully it will inspire you to look in your textbook, look up some vocabulary words, and maybe review some topics so that we can pull all this together and understand the kingdom to which we belong. We're going to focus on three topics about animals. We're going to look at the diversity of the animal kingdom, which is enormous and very familiar to you. A little bit about the evolution of the animal kingdom from simpler organisms that have been around since the Cambrian explosion. We're going to look at body plans, basically how are animals laid out, how are they put together. And finally, we're going to focus on animal body systems, look at some systems specifically. This video cast is not going to cover animal body systems. That will be the second part. All right, so the first question that we need to answer is what exactly is an animal? What makes something an animal? This goes back to basic biology, but all animals in the animal kingdom are multicellular, okay, which means that they contain more than one cell. And now all animals develop from a zygote, which started out single-celled, then goes through mitotic divisions, of course, to make lots and lots of identical cells, which start out all the same, and then differentiate into all the different tissues and organ systems that we all know and love that are part of animals. The other thing that makes animals animals is that their bodies are heterotrophic, which means that they do not have the ability to make their own foods from carbon dioxide and water, like we see in the autotrophs, more commonly known as plants and algae. And finally, and probably the least familiar to you guys, is that all animals develop from three basic embryonic cell layers. And I'll show you some pictures that will hopefully set you on a good course if you want to explore that further. All right, first of all, excluded from the animal kingdom are the unicellular heterotrophic protozoans that you may know of, for example, as Paramecium and Euglena. These are the organisms that were first discovered when early microscopes were developed in the 1650s, including the work done by a guy named Anton von Leeuwenhoek, who called these guys animalcules. In this picture, you can see some, this is a cartoon from the period, based on some drawings that he published. A lot of what he saw under the microscope were in fact the larval forms of lots of multicellular animals, but he also saw mixed in with these guys lots of paramecia and protozoans like amoeba, euglena, lots of swimming small plants and animals. He grouped them and called them animalcules. Those are generally excluded from the animal kingdom and are shoved into a kind of a catch-all junk drawer kingdom called Kingdom Protista, which also includes the photosynthetic or autotrophic ones that you guys probably know as algae and seaweed. Not a good way to organize a kingdom, but that's a topic for a different video cast. All right. All multicellular animals in the animal kingdom are heterotrophic, which means that they have to eat. And when you eat, you satisfy two basic cellular functions. Okay, you bring into your body organic molecules that your metabolism is going to need to build all the different body parts, tissues, cells, whatever that you need. And this is all part of that, that building process called anabolic metabolism, which is the building part. Also, when you eat, you bring into your body calories that your body is going to be using to make ATP. And remember, ATP is the basic molecule that runs all the functions or all the metabolic pathways that keep cells going. Now, you need to know what these organic molecules are that are used as building blocks. So please review that if you can't recall those. And you also need to know something about how ATP is made. Again, we studied this earlier. Please review it if you think you need to refresh your memory about this. Okay, the embryonic layers bit. All right, when a zygote starts dividing and becomes multicellular, and it starts out as one cell, of course, and becomes a ball or a collection of many cells. These many cells, which are called stem cells early on, quickly start differentiating into what are going to become what we call embryonic germ layers or embryonic layers. There are three of these. The first layer of cells are called the ectoderm, and they form the outside skin of the developing embryo. Then the endoderm forms inside of that ectoderm layer, and that forms second. And then in some animals, they develop a third layer called the mesoderm layer, which is the last layer that forms. It's going to be sandwiched between the ecto and endoderms. The ectoderm becomes the outer covering, as I said. The endoderm becomes the digestive system, all right, or which is basically a tube if you think about it, that runs from the mouth all the way to the other end. And the mesoderm, which not all animals have, can become different things. Again, the point of this video cast is not going into a lot of details, but just make you aware of some things that you really should explore further in your textbook. All right, the development of these three layers, when they form and what they become, is a huge clue to the phylogeny of the animal kingdom. It's studying these embryos is how scientists have decided who was related to whom at really, really ancient Cambrian explosion time period history. Okay, so these differences in these layers is a really basic and big clue to the phylogeny of the entire animal kingdom. Again, we're not going to go into that in this video cast, but there are sections in your book which show this in beautiful detail. Take a look at it, see if you can figure some of this stuff out. All right, so we're finished with the idea of the different kinds of animals and their evolution. Now we're going to move on to body plans. Okay, all right. In the animal kingdom, roughly, there are two different ways of evolving, all right, from this original zygote. You can either become an invertebrate or a vertebrate. And I know you've heard these words before. Invertebrate body plans basically are hard on the outside and soft on the inside. And there is not a structure that runs down the back or down the belly that is a stiffening rod of any form. Invertebrates can have a radial or a bilateral symmetry to their body. We'll talk more about that later. Vertebrates, on the other hand, are kind of inside out. They are hard on the inside and soft on the outside. They have a stiffening rod down the center, and they are only bilaterally symmetrical. All vertebrates have a bilateral symmetry. Now, you may have figured out that what we're talking about hard on the outside, that's an exoskeleton. The classic example of that would be like a lobster or shrimp or an insect. And of course, vertebrates have an endoskeleton, like, obviously our human skeletons. This stiffening rod down the back, in humans, of course, is a vertebrate, which is is a vertebral column, which is what we get the word vertebrate from. But in other animals, they don't develop that far, but they develop something called a notochord, which appears in embryonic vertebrates, but then becomes something else. So please look up a notochord, make sure you understand what it is. But again, it's one of those clues to the phylogeny of vertebrates, how they're related to each other. Now, for symmetry, what we're talking about here is what's the overall shape of the body. And in the case on the left here, we have basically no symmetry, which is how sponges develop. Sponges are the simplest of all the animals that make it into the animal kingdom. In the middle here, you see radial symmetry, which is like a sea anemone, a body basically based on a circle. No head, no tail, just a top and a bottom. And finally, all the way to the right, we have the classic bilateral symmetry, which we see in all vertebrates and all insects and a lot of the familiar animals that are running and jumping and flying around. And you have a distinct head and tail and a distinct left and a right. Okay, and a top and a bottom also. So lots more geographic orientation type words in bilateral symmetry than in radial symmetry. Sea anemones are basically circles with a mouth in the middle. All right. Now, organisms that don't have any symmetry like sponges tend to develop and grow in one place. For a long time, people thought sponges were actually a weird type of plant, but they're definitely animals. The word for not moving around is sessile. So that once they start growing and developing, they're locked in one place. They don't swim, they don't crawl, they don't move. If you are radially symmetrical, because you don't have a set of sense organs concentrated in a head, you can move like starfish and sea anemones. They all move, but they tend to be slower moving. They're not known for, or I can't think of any example of a fast swimming or fast crawling radially symmetrical animal. If you want to be a predator and a hunter, fast swimmer, you're going to have bilateral symmetry. And that's what we see in the part of the animal kingdom that includes fish and amphibians and reptiles and birds and mammals. Okay? All right. We have finished body plans. Now, the second part of this video cast is going to talk about animal body systems. And I don't know how many body systems I'm going to be able to fit into each one. So we'll see how that goes. But thank you for listening, and we will stop there.